Good morning or good afternoon, everyone, depending on where you're located. My name is Zsuzsanna Hollander, and I'm the Director of Data Science at Genome British Columbia. And while we meet today on a virtual platform, we would like to take a moment to recognize and acknowledge that we live, work, and raise our families on the traditional territories of Canada's Indigenous people. We do this to reaffirm our commitment to and responsibility for improving relationships between nations and to enhance our understanding of local Indigenous peoples and their cultures. Genome BC is situated in Vancouver, British Columbia, which is in the traditional territories of the Coast Salish peoples, including the Musqueam, Squamish, and Sabletooth nations. And with this, I would like to welcome you to Genome BC's Data Literacy Webinar Series. We started this last fall, actually exactly a year ago, with the aim to bring national and international experts together to help deepen our understanding of different data-related initiatives, standards, and resources. Today's presentation is part of a mini-series we started in September focused on artificial intelligence. It is my pleasure to introduce our speakers, Casey Shannon and Linda Lapp. Casey, I think, is a data scientist rock star, even though he'll probably deny that. I had the great pleasure of working with him for over 10 years, and his work in the translational research arena has been outstanding. He always brings a fresh perspective to any project that he works on and any data set. He is innovative with method development, embraces new omics technologies, and is thoughtful in the analytics that should be applied to each. As a result, he has been sought after in the biomarker development community and is always working on many exciting and uniquely challenging projects. Linda Lapp is a statistician and data scientist interested in predictive modeling and its applications in healthcare. In fact, she's coming from the UK, where she has done quite a bit of clinical research, and her PhD work involved analyzing complications such as acute kidney injury and delirium following up open heart surgeries. And so both of Linda and Casey are coming from the Proof Center of Excellence, and with that, I'll hand it over to them.
Thanks, Zsuzsa. A bit embarrassing. It's an embarrassing introduction, but thank you for the kind words. So I'm very pleased to be here. I'd like to thank the organizers for giving me the opportunity to really do a bit of reflective work, because this was or is to be a presentation on pretty much the last 10 to 15 years of work here at the Proof Center. Some of it predates me, so if I get some of the details wrong, you can ask Zsuzsanna, who we used to work with, and so some of this is her work, and she'll correct me if I get some of it wrong. But I'll be talking today about one of our oldest programs called HEARTBiT, which is an effort to try and develop a heart rejection diagnostic test using genomic technologies. There we go, I'm moving forward. So first, I'd like to go over some of the learning objectives, because this is, after all, a data literacy series. So we're going to talk about common types of genomic biomarkers today. We'll outline some of the important steps in development of that kind of diagnostic. We'll talk about a few of the machine learning methods that are relevant to biomarker discovery in this space, although it'll be pretty light on the details. So I apologize if you're here for the details. There's some more details towards the middle bit. And then we'll discuss some of the limitations presented by tissue complexity. A lot of it is going to be blood focused. I hope you like blood. It's a bit of a weird I hope you like blood. And then we'll briefly discuss some of the opportunities offered by data integration in this space. So first, I'd like to start with something not omics related at all. This is Dr. Jonathan Swift. He's a writer, and he wrote a piece, a satirical piece, a long time ago, called or titled "A Modest Proposal for Preventing the Children of Poor People from Being a Burden to Their Parents or the Country, and for Making Them Beneficial to the Public." This was a satirical essay that Dr. Swift penned, aimed at trying to solve the problem of extreme poverty in Ireland. And in this essay, he proposed a bit ludicrously that really poor Irish people should sell their children and they could be used as food to feed rich British people, which was obviously written in jest. But the reason I introduce it here is because I'm going to adopt, or I'm going to propose a similarly ludicrous proposal, or maybe ludicrous to the people on this panel, that you should engage in clinical research, even if you're interested in sort of heavy data method development kind of work. And so that might sound ridiculous. Maybe you think you want to go into a pure sort of theory kind of context to do that kind of work. I'm going to propose that clinical work and clinical research is an ideal venue to come up with some problems that need to be solved for very important reasons. And so they foster a lot of innovation. And I think that the 10-year sort of overview of this HEARTBiT program will be a good illustration of that fact. So that's Mr. Swift. I'm not going to continue in that satirical sense because I'm not as good a writer as Dr. Swift. But so briefly, the Proof Center, I think Juja did a good job of introducing us at a high level. Proof was established back in 2008 as a Center for Excellence in Commercialization and Research, or CSER, which was a program of the Canadian government at the time that gave us quite a bit of money to establish ourselves. And initially, our programs were centered around prevention of organ failure. Proof stands for prevention of organ failure. So we started with a number of programs centered around transplantation. And today I'm going to talk to you about one of those programs. But since then, the scope of the work has expanded quite a bit to include a number of chronic diseases. And those are listed on the right-hand side of the slide. We don't really have to get into the detail. We've worked in a lot of different indications over the years. So we have varied interests. But today we talk about transplantation, which is sort of our origins. So heart transplantation. Why are we talking about heart transplantation? Heart transplantation is the only treatment for end-stage heart failure. And acute rejection, which are effectively the body rejecting the new organ, are better controlled than they used to be because we have good immunosuppressive drugs. But nevertheless, rejection remains common. About 20 to 30% of heart transplant recipients experience one or more episodes of acute rejection in their first year post-transplant. Those episodes are associated with increased risk of morbidity and mortality and possibly loss of the organ. So obviously, they're important to catch and possibly intervene early if possible. Currently, they are monitored for using the endomyocardial biopsy, and that's pictured sort of rudimentary in a rudimentary fashion on the right-hand side of the slide here. But effectively involves sort of a surgeon slipping a small catheter through a vein in your neck, pushing it into the heart proper, and then nipping a piece of the heart tissue so that tissue can be put under a slide and look. A pathologist can look for evidence of immune cell infiltrate. There's a lot of these in the first year post-transplant, 12 to 15. They're, you might imagine, not particularly comfortable. They're done not under general anesthesia, so not a great experience, and you have to go through 12 to 15 of them. They're also quite expensive to the healthcare system. So HEARTBiT as a program was really born out of the need to address this issue, right? So rather, we wanted to replace this sort of invasive surgical procedure with something far less invasive. And the proposal was to go after to try and develop a heart or a blood-based molecular marker to diagnose acute rejection. The first few texts or first few slides are text heavy, but we move into more diagrams, I promise. These are sort of necessary. I just wanted to define some terms that I'm going to be using quite a bit. Biomarker is one of them, as you might imagine when describing a biomarker program. So what's a biomarker? Well, it's a thing. It's a thing that you can measure that reflects either a normal biological process, a pathogenic process, or response to an intervention or an exposure. That could mean, environmental exposure. It could also mean a drug, or it could mean a surgical intervention even. Biomarkers could be a lot of things, but in our case, we're talking about molecular biomarkers and specifically genomic biomarkers. But of course, you could derive biomarkers from other types of features, and we'll introduce some of our more recent work along those lines towards the latter end of this talk. And I think one of the interesting things, so that this was a working group paper that was published in 2016 by the FDA. And one of the things that they introduced that was kind of novel at the time that they published it was that it's not enough to just describe what you're measuring and what you think it correlates to. You should also describe the biological plausibility and how you measured it, right? So it's not just about saying, I measure this protein and I think it correlates with an outcome. It's about, okay, but specifically, how do you measure this protein? And why do you think that this is relevant? It's not, and I think this was a reflection of issues that the FDA working group identified in some of the research that was coming out in this sort of genomic high dimensional assay space around diagnostics, which we will discuss in a minute, because I think we made some of those same mistakes in our early work. Briefly wanted to discuss types of biomarkers. So you can use these signatures for a lot of different things. We're mostly going to be talking about diagnostic biomarkers, so markers that are created to detect or confirm the presence of a disease or condition, or possibly to identify disease subtypes. That we'll get into again towards the later end of the talk. But we'll also touch on susceptibility risk markers or prognostic markers. So these are markers that are trying to identify sort of baseline susceptibility to developing a particular disease or a complication of that disease. So there are those are the types that we're going to be discussing here, but there are other types like monitoring biomarkers, predictive biomarkers, biomarkers of response, safety, that sort of thing. So HEARTBiT. HEARTBiT, as I alluded to a few slides ago, is a program to identify a minimally invasive, so blood-based genomic biomarker of acute cellular rejection in heart transplant. I'm going to come back to this slide a couple of times during the talk, but it lays out sort of the different aspects of the work that I'll be introducing and discussing with you. I modeled this slide actually after something our group published back 10 years ago now. Time goes by. But at the time, we thought we'd sorted this out. So 10 years ago, we put out a paper that sort of described the major steps in developing of a genomic or sort of omic in general diagnostic test. And we highlighted these sort of three main phases that I'm showing pictorially here at the top of the slide. Roughly speaking, the identification of candidate. This paper was written specifically about proteins, but in this case, we're going to be talking about candidate transcripts or genes from a large pool. So, even back in 2008, you could measure, all of the genes, the expression of all the genes in a tissue sample. And the discovery phase, the job was really to find a handful of relevant genomic features from that big pile of data that you generated for your initial experiment. And then we envisioned that the following step was really to take those candidates, move them to a more clinically amenable platform, And there were sort of specific concerns for proteins around really sort of like pragmatic concerns, right? Instrument time, cost, ability to sensitivity in a multiplex kind of context. Maybe less relevant in the genomic space and maybe less relevant today than it was 10 years ago. But that is sort of the paradigm that we followed in our work with HEARTBiT. So there was a translation piece to a different platform, a targeted platform that could be run at a lower cost and possibly point of care, if that's sort of an important consideration. And then once that work was done, also involved some replication in new samples. Then we would move into the implementation phase. So back in 2013, we had all this sorted out, at least in our head, but we were still very much in the discovery phase back then. So we'll start by describing a little bit of the work from that discovery phase before discussing some of the latter work around validation and then moving into implementation, which is what we're currently working on. So heart rejection, I'm going to try and fly through this this early discovery work because it's going to feel dated, I think, for some people. This is, you have to remember sort of some of this was started 15 years ago. Very different times, right? So one, the platforms were different. I'm going to talk a lot about microarrays in the first 4 slides or the next 4 slides. That might seem quaint, but they were the workhorse of genomic research back then. I'm also going to talk about studies that, the numbers are going to look quite small, and we're going to talk about why that is and how that was a challenge. But let me briefly take you through some of this early work that set us up for the latter work that we're currently engaged in. So we had this first paper in 2009. We'd been enrolling as locally in Vancouver for about 4 years at this point. And we had a bunch of people who had undergone heart transplant that we'd collected blood from, both at any of the protocolized biopsy time points. So after heart transplant, I mentioned 12 to 15 biopsies. They come in sort of very frequently in the early time points and then sort of progressively less and less often. Every time a patient would come in for a protocolized biopsy, we would get a blood sample. And so we had a pathologist certified, yes, this is rejection, no, this is not rejection, or maybe in between, for all of these time points, and we also had blood. We went ahead and profiled using Affymetrix microarrays a relatively small set of patients, although I'll point out that there aren't too many heart transplants nationally in Canada. There are obviously less just locally if you're just looking at Vancouver. And so accruing samples is sort of a time-consuming process. So we worked within those constraints. But nevertheless, we had a small set of participant samples that we'd managed to accrue over the last couple of years. We had some acute rejection events, and so we went ahead and profiled this relatively small set in sort of modern terms, relatively small set of patients. Measured all of the transcripts from their blood, either at the time of rejection or during a non-rejection time point. And then we looked for genes that were different between those two groups of individuals. I'm highlighting a few methodological details. They're going to seem kind of out of the blue right now, but we'll get back to them at the end of this discovery step, or this section described in the discovery work. One of them is lemma. So this is a moderated t-test, sort of very high level. We basically the estimate of the mean difference between the groups or whatever is shrunk towards the mean. I'm missing the details, but sort of to give you an idea, rather than just sort of trusting the estimate of the fold change, say, between the groups. If it's a very, very high value relative to all the observed values, we're going to shrink it a little bit towards the mean. If it's a very low value, we'll shrink it sort of up towards the mean. Although, given that we're measuring all of the genes, the mean's probably going to be pretty close to 0 because most genes are probably not going to be different between the groups. So this is what we were using at the time. It was slightly more sophisticated than just a simple t-test. We identified a bunch of probe sets, so it's not even really transcripts at this point because these are microarrays. So they're these sort of targeted oligonucleotides. We're measuring these groups of targeted oligonucleotides. We identified these probe sets, and we build a model. And this model, it's maybe small to see on this slide, but in the lower right-hand corner, we report various performance metrics on this model. It did very, very well in cross-validation. We'll talk about why that's a bit misleading when we do the recap at the end of this section. And then we had a small set of samples that we kept out that we didn't use to train, and it seemed to work in that small set. And so we were quite pleased with this result. And that was our first study. We had sort of demonstrated that maybe there are genes that are different in blood when we compare rejection time points to non-rejection time points. That was in a very sort of idealized scenario. So we were using what at the time we called these extreme phenotypes. These were uncomplicated cases, right? Clear-cut rejection, clear-cut not rejection. It was their first rejection, not their third, right? Because people tend to reject in clusters, or there are like sort of people that will reject more than once, and once they've rejected once, they're more likely to reject again. So all of that complexity was out of there. We weren't including it in any meaningful way. This was a follow-up piece of work actually by Zhuja, looking at trying to include some of these more complicated cases. So we're looking at a sort of a slightly expanded set of samples from the set that we were discussing previously that now includes these complicated cases. And we're not going to go into the details of this one, other than to say that the methodology had evolved a bit by then. But really the sort of salient thing about this study was that once we started including these complicated cases, our ability to discriminate between acute rejectors and non-rejectors looked a lot worse. So the panel on the lower right-hand side here is an area under the receiver operator characteristic curves, an AUC. It's labeled D on the slide. And you can see that it's sort of very close to the diagonal, which is effectively equivalent to a coin toss, right? So we've got a signature, but the ability to discriminate between rejectors and non-rejectors once we've got these complicated cases in is not great. And Zhuja did a very clever thing here. We knew, of course, that the biopsy, the tissue biopsy specimen itself probably had a lot of relevant information with respect to rejection, after all, where the process is happening. And so we, or Zhuja rather, leveraged that biopsy to winnow down the list of genes and build a model from those genes, and was able to derive a useful signature with that strategy. I think we're going to skip this one, other than to say that blood is a complicated tissue made up of a lot of different cells, and we did a bit of work to try and understand how the changes in the cellular composition of the tissue at the time of rejection and at the time of non-rejection affected our ability to detect differentially expressed genes. So that was a bit of method development that we did specifically because of the complexity of the tissue that we were using to derive the signature. And then sort of yet another strategy to identify genes related to rejection. This was trying to a very similar study to the one we just talked about, also by Zhuja, but now looking at pre-transplant. So are there molecular things in blood and in the tissue that is being transplanted that can predict whether someone is going to reject or experience episodes of acute rejection or not? Again, it was a sort of trying to combine the biopsy with the blood readouts, and she was able to demonstrate that whole blood alone doesn't really carry information that is predictive of rejection, but if you focus in on genes, that are driven by the tissue itself, you are able to derive a signature that might be useful in a predictive context. So all of this work kind of came to a head. We sat down, we had a lot of lists of genes from these various studies, and we knew that we had to move some of them, a selection of them, to a different platform to start working on this next validation phase. And in the end, it's sort of a little bit of each of those studies made it in. So we sort of sat down, we wrote down all of the genes that we thought were, that we'd learned were relevant to the process of rejection in blood. And we translated some of those to a targeted platform, which I'm going to talk about in a second. But I wanted to sort of highlight a few of the things that this phase of work taught us. So one, these are all very small studies, right? To be fair to ourselves and to some of our past members who did some of this work, enrolling is hard, especially in heart transplants. So that I mentioned, there's not too many of these that happen every year in Canada. There's even fewer when you're working at a single site. Enrolling patients is hard. That's one of the challenges of clinical research. At the time too, running these genomic platforms was expensive. Nonetheless, there were a few learnings from this phase. I think one of the big learnings, and we sort of revisit this periodically with different collaborations that we engage with, because this is something that people still get wrong. The importance of proper cross-validation, right? So doing it the right way to get a sort of accurate estimate of the generalizability of a model was something that we learned very early on in this work. The other sort of methodological insights, so I mentioned lemma, I didn't mention the lasso, but some of the biomarker work that I presented in Juja's papers we're using a penalized logistic regression to derive a signature, to ultimately derive a biomarker signature at the end. And that penalization is the lasso. Both of these introduce a conservative bias to our estimate of the association of a particular gene with our outcome. And both of those were important methodological improvements to the work that we realized in that discovery phase of the work. And then finally, and this is sort of a preview of what's to come, but leveraging external data proved to be extremely useful, right? So we introduced complicated cases, there's no signal. We leveraged the biopsy to drive the identification of relevant gene features. We're able to derive a signature and so on. So we leveraged that kind of thinking a number of times in those those first few years of work. And it was a preview of things to come, because this idea of integrating across multiple, in this case, it was tissue modalities, but in the future, we eventually ended up integrating across multiple molecular modalities as a useful strategy to derive better models that generalize better to new data. And so that was, we didn't know it at the time, but that was an early insight. So now we had all of this sort of imperfect work. Admitted, we knew this at the time too. It wasn't what we wanted. We would have loved to have 1,000 acute rejectors and 1,000 non-rejectors. We didn't have them. We still had to move forward with the work. And so we, as I described, sat down, wrote down all the genes we thought were relevant, moved some of them to a more clinically amenable platform, which was the nanostring platform. So I don't really have time to go into what's so special about this platform. Suffice to say, it's a targeted platform. We had to design capture probes for a set of genes that we had determined were of interest. So we did that. And then in the background during this discovery phase, we had continued enrolling. So the nice thing about the second part of the work is that by now we were multi-site, right? So we were enrolling in Vancouver, but we were also enrolling in Toronto and Ottawa and in Nebraska in the US. And or actually, sorry, no, for this one, we were just Canadian at this point. But it was a multi-site study by then. So we were accruing samples a bit more rapidly. And also there's this sort of additional robustness about getting sort of varied samples from different parts of Canada. So this work was basically to confirm that the set of features that we identified in the prior part of the work were relevant to acute rejection. So we designed this assay. We ran it on a whole new set of samples. We nearly doubled the sample sizes of our early studies. We were looking at approximately 36 acute rejectors in total, and nearly 86 non-rejectors, 86 non-rejectors. And then additionally, we were also, although we didn't use them to train the model, we were also trying to look at whether the signature was able to discriminate between mild rejectors. So these are these sort of in-between cases that have evidence of rejection, but not enough to modify therapy. And so importantly, up to now, we had never looked at these, right? The signatures were derived to try and discriminate acute rejectors from non-rejectors. It could have gone a lot of different ways, but sort of fortunately for us, the set of genes that we had selected performed in the way that we wanted it to perform. That is to say, identified acute rejectors and was able to discriminate those from both mild rejectors and non-rejectors, which clinically speaking are equivalent. There's no modification to therapy in either of those. So we think of them the same way. And so we did this work. We used a little bit of the data to do recalibration. We tested the signature in a second set of samples. And then we also assessed whether it held up when including these mild cases. And it worked. So the performance wasn't as good as what we were seeing in the discovery phase, but I think that was expected. And importantly, we had a signature that, given a sufficiently strict cutoff, could predict or could discriminate rejection and spare approximately 50% of protocol biopsies, which is quite significant. We went on to do some analytical validation work. So it's pretty dry stuff. It was really about demonstrating that we could reliably measure these genes across a range of RNA inputs, sample quality, and that sort of thing, across batches, across runs, across days. So all of that looked great. One of the learnings from this particular phase of the work was around timing, so modifiability of the signature. So far, all of the studies have been these cross-sectional studies. We didn't really know whether the signature represented inherent risk or whether it was a modifiable sort of thing that went up at the time of rejection and then down after. So we demonstrated that, yes, it was modifiable. And then we also further validated its performance in another set of 16 rejectors and 16 non-rejectors. Very, very similar performance to what we'd seen previously. So in the background, we'd been dealing with this. I introduced this sort of problem, this issue that we were coming up with in the discovery work. We sort of, we had one study, it looked good. We introduced complex cases, it doesn't look good. We leveraged the tissue, it looks good again. Clearly selecting the right genes that were generalizable across sort of varied populations of heart transplant patients was challenging. And it's really not a surprise given that we're working with so few patients. I'd introduced this idea that introducing conservative bias to our estimates of association of genes with outcomes was useful, and that leveraging external data was also useful. Both of these things are evidence of overfitting, right? So we're giving these models very, very long lists of features and very, very few samples. This is this sort of N much smaller than P context of far fewer samples or observations than variables. And in that context, you can find features that perfectly distinguish between any arbitrary grouping of your sample, of your observations, which is problematic. It makes finding relevant genes difficult. And that problem is sort of fundamental to these platforms and in the clinical research context. We're not going to get to a point where we can run these studies on 10,000 patients. So this was a problem that we wanted to solve sort of concurrently to do this validation work. This is a tweet I like from Martin van Smeden. It's something that's a lot of people get wrong. So he reminds us that cross-validation or bootstrap is for the diagnosis of overfitting, not its prevention. So you can do all of the things that we had been doing to make sure that you get a robust estimate of how well a model will perform in new data, but that doesn't change the fact that the method that you're using has a tendency to overfit. So it's not fixing the method. So how can you fix the method? In 2019, we published this paper. It's a method that we had developed. I was secondary to this. This is work that was led by Amrit Singh, who's a young early career faculty at our center now. Method was called Diablo. It was a multi-omic integration method. Conceptually, I'm going to fly over the concept, but basically, it was trying to leverage the fact that systems of cells are regulated at different levels. Transcriptionally, translationally, post-translationally, et cetera. And we propose to basically leverage our ability to measure all of these simultaneously in the same set of samples to try and identify better features, more generalizable features, and basically reduce the identification of false positives. I'm going to skip the math, other than to say that, well, we don't need to get into the math. It's basically in the family of methods called PLS, so projection to latent structure. It's analogous to principal component analysis, except it's driven by a label. It's a supervised method. And the work that Amrit did in that paper was basically to extend it to multiple matrices, multiple molecular inputs, and relating it to a discriminatory type of exercise, so to build a classifier effectively. And so in this work, we showed that this integrated methods did just as well in identifying sort of features that are able to discriminate between two classes of patients. So here we were looking at various sort of cancer phenotypes, people that survive a long time, people that don't have very long survivals, and trying to identify features in blood across a number of molecular modalities that can identify those that survive from those that don't. Diablo performs sort of equivalently to other supervised methods, but it had this interesting property that when you compared the performance in training, which I'm showing in red here, and the performance in new samples in a test set, so two things. One, its training performance typically looked worse, which you might think, well, why would I want a worse performing model? But in effect, it reflected the fact that it was working under heavier constraints, right? Because it was trying to select sort of congruent features across multiple molecular modalities. And so the sort of consequence of that is that it had much less tendency to overfit. And I'm depicting that here by sort of the shaded area, which is the distance between the training performance and the performance in unseen samples. So it sort of had less of a tendency to overfit. It also often resulted in better performance in the test, so more generalizable signatures. A consequence of that, which is sort of like not directly, but we think is a consequence of this lower tendency to overfit, and so selecting sort of non-false positives, like actual genes and proteins and metabolites that are associated with the outcome, is that the features that it selects tend to line up much better with annotated gene sets. So when you sort of like look at the proteins and genes that Diablo was selecting and you compare them to large collections of annotated gene sets like molecular signatures database, that sort of thing, the features that Diablo pulled out were sort of consistently enriched for a lot of known pathways and annotated gene sets. So that's sort of like saying, well, you know, really, if in a vacuum, if we were selecting a bag of 100 genes that we thought were associated with the disease, we would hope that it would line up with what we understand genes to do under various circumstances. And so we would expect them to line up with established biology. That was sort of in the original publication. We went on to show that that kind of thing happens in other contexts. So this was another piece of work looking at the developmental pathways of neonates in the first week of life. And we published that work in Nature Communications in 2019. Similar conclusion there. So, you know, that was sort of just to highlight an opportunity to do a bit of method development that really came up as a result of like challenges that we'd identified in carrying out sort of basic clinical research. Next, I wanted to talk about some of the stuff that we're currently engaged in. So I'll fly through this because I have like 7 minutes or something, and I want Linda to talk about a few things to you as well. So we've moved in now to this implementation phase of HEARTBiT. And that's largely this sort of prospective trial that is ongoing. Effectively, it's going to be make or break, right? We've got our signature, we've got it developed for this nanostring platform. We've got analytical validation that shows that the analytes perform well under various conditions. And now we've gone and prospectively enrolled a number of patients across these sites. We've got all the samples, we've got all the labels. We just have to run the assays and crack open the signature to see how well it works. So we'll find that out early next year, probably. Yeah, I'll let you know. In parallel, there's a few sort of issues that have been plaguing this development effort that we've decided to do sort of additional work on. One is specificity. So, you know, I mentioned, I have this high-level comment about HEARTBiT. We could spare roughly 50% of the biopsies. That 50% number is really down to the specificity of the assay. We can be extremely strict about what we call a rejection. And then we often, when we say it's not rejection, often we will be the other way around. When we say you might have rejection, we're going to err on the side of caution. And so we might say you have rejection even if you don't, and though you still have to go and do the biopsy. So improving the specificity is something that we're very much interested in doing. This was work that was just published earlier this year, looking at trying to integrate a signature of T regulatory cells to see whether it could improve the specificity of the signature. It looks like it might a little bit, although there's this, seems like Tregs are kind of like more of a prognostic or susceptibility kind of marker. Like rejectors, if they had rejection, they seem to be high on this Treg signature, regardless of when you measure them, which is kind of interesting. So that's kind of TBD work that we're trying to push forward to try and shore up the specificity of this signature. The other thing that we're trying to fix is this sort of big challenge around the labels. So all of our work is grounded in pathology-derived labels. They are the gold standard for determining whether there's rejection or not. They're terrible. I mean, if there are pathologists on the call, I apologize. But it's not just me saying it. We ourselves have experienced a lot of very poor agreement between the expert pathologists that we asked to review our slides to determine our labels. It's also sort of widely reported in the literature. And that's a challenge because all of the molecular data, you know, you can have all of this molecular data, but if you're building a surrogate in blood against labels that are wrong, and I'd argue that because pathologists disagree with each other, some of them have to be wrong some of the time. So that's a big challenge to overcome. We're sort of pursuing this on two different fronts. One is adopting some ideas out of a group in Calgary. This is Phil Holloran's work. It's been going on for ages, but they've done quite a bit of work trying to basically contrast histology-derived labels, i.e. the stuff that a pathologist is looking at, with molecular profiling of the biopsy itself. And I'm showing that in the PCA plots in the middle of the slide here. There's very little agreement between the molecular picture of the data and the histology labels. And so they sort of propose that really we should be looking at this molecular readout as the ground truth rather than what the pathologist is saying. So that's something that we're exploring with single cell and spatial genomic technologies. And then in parallel, we're also trying to improve the inter-rater agreement, and that's largely being done using these sort of newer deep learning approaches to mostly create sort of a pathology assistant, right? So it's the idea is to highlight areas of a slide that a model thinks are relevant to rejection based on a large set of training data, so that the pathologist can sort of look at the right areas, and hopefully, if they're all working under this system, their agreement would improve, and that would lead to more consistent labeling. And so with that, I'm going to pass it along to my colleague, Linda, who will talk to you about, well, I'll let her tell you what she's talking to you about.
Thank you, Casey. Oh, okay. So I'll just a second. Okay, now it should be good. So thank you very much, Casey. So I'm going to give you a brief overview of a different project that is connected to the whole HEARTBiT program that is looking at outcomes beyond acute cellular rejection that mostly HEARTBiT is all about. So this project is called Data Access Integration and Analysis Program, also called DIA, and it's kindly funded by Genome BC, which we're really grateful to work together with on this project. Next. Oh. Okay, so for heart transplantation, essentially a person is getting a foreign object into their body, a new heart, and therefore it is kind of difficult for the body to accept it. And because of this, the patients are given immunosuppressive medicines to avoid rejecting this foreign object into their body. So currently, the protocol is to use the same approach to each patient. There's no personalized approach to this largely. So if a patient is given a lot of immunosuppression, they are less likely to reject their new heart. However, if they're given too much immunosuppression, they are likely to get some other complications that could occur in the long term mostly. So for example, so it's a balancing act here. So if the patient gets too much immunosuppression, in the short term, they're likely to get some infections, which could be cytomegalovirus or Epstein-Barr virus, which are two very common complications for transplantation patients. Also, chronic kidney disease in the long term because of the kidney toxicity. And in about 5 years or so, patients are also likely to get cancer, most commonly skin cancers and gastrointestinal cancers. So it's really a balancing act. And what we are trying to do with this project is to look at how we could predict these different complications and potentially diagnose so that we could optimally titrate the immunosuppressive treatments for the patients. So ideally, they would get to live their lives in a more quality manner. So the DIA project essentially is using this new tool that was developed by Providence Research and Providence Ventures. It is called Integrated Health Informatics Data Lab, called IHEAD. And what it is essentially is that they are taking the data from different electronic health records and they are adding them into this one environment where the researchers could access this data so that they could answer their research questions. So in this project, we are looking at different diagnoses, routinely collected laboratory results, medication data, and imaging data, so that we could link our data from the HEARTBiT project and look at these outcomes. So we can see also beyond the acute rejection patients. So far we have some results to understand what is going on in this population. So the most common complications are obviously the cardiac complications, which is 11% of these patients that we have in this database. Also renal complications, diabetes, and different kinds of infections. And on the left-hand side on this figure, we can see that as the years go on, the more and more different patients would get these complications. So essentially by year 6 or so, about 10% different patients would get these kinds of complications. So we have done the preliminary analysis regarding what kind of complications there are. So our next steps is to use the nanostring technology to understand, to get the transcriptomics data and the Treg data to link with the clinical data that we have from the IHEAD database. And then ultimately we are looking to see which biomarker signatures are associated with these different kind of complications so that we could predict these earlier and also potentially diagnose these.
Okay, I'm back. And so, okay, this is a bunch of people that were also important to this work. I pulled older pictures, so we all look younger and we all have hair. And I just turned off my video camera, so you also saw that at some point I also had hair. So yeah, all of these people were sort of crucial to this work. And I would like to thank them and these funding agencies and you for your attention. And with that, I guess we'll take some questions.
Thank you, Casey and Linda. I also wanted to acknowledge that the Proof Center has agreed to post their metadata on our Health Omics metadata commons that we're just creating. So thank you very much for Casey and Linda and to put together the metadata so that prior to us launching the metadata commons, we have a few data sets in there. And also would like to welcome anybody who's on the call who has omics data sets to please reach out. We would love to work with you to share your metadata on our commons. And so with this, we'll go to the questions. We have a few minutes. One of the questions is, I think both the questions that I see right now are interesting from very different reasons. How many of the markers are applicable to other transplants? For example, kidney is one of the questions.
Yeah, that's a good question. I actually, maybe the person asking the question might know, but we had a large program in kidney. It was actually it predates the heart program. It was not pursued for various reasons, but in terms of the overlap between the features that we're talking about here for HEARTBiT, I don't know, to be honest. I don't think that we've systematically done that comparison as part of the early work, although it could have been done at some point. I'm just not aware of it. For the signatures, the sort of the genes that ended up on this panel, the 12 genes that are part of the HEARTBiT signature that has been moved forward, we haven't run that panel in a kidney transplant context, so I'm uncertain. It is sort of, I guess, compelling to think that these processes like solid organ rejection would have some commonalities, and there are definitely groups that have looked at sort of the the pan organ rejection, although I don't know that there are a lot of success stories on that front with this kind of approach. There is the sort of circulating donor DNA angle that is sort of, or looks to be, if you look at the CareDx product, looks to be a pan solid organ solution, maybe. Yeah. But thanks, that's a great question.
Yeah, I think I remember at some point we looked at it and it was strangely, whatever was upregulated in the heart transplantation was downregulated in the kidney transplantation, but you know, this.
Oh, okay. I mean, sort of maybe another thing to highlight, and this is, again, if there are doctors on the call, I apologize because I'm going to mess this up, but the sort of major processes that are driving rejection in the two organs are not always the same. So in heart, there was a big focus on acute cellular rejection. Antibody-mediated rejection is catching up, or is becoming more of a thing that people are studying. But back when we started this, it was really not on the radar, wasn't thought to be all that important in heart, but is much more relevant in kidney. And so that could be a discrepancy as well in terms of the molecular features that are implicated or associated with rejection. Just the physiological process of it might differ between the organs.
So the next question is related to the pathologist rating of the biopsies. One is, can AI be used to screen prior to asking the pathologist what the biopsy says?
Yeah, I mean, probably. I think you do. I mean, you know, I talked about it a little bit. So we do have a bit of a small program around trying to create an augmentation to the pathologist using an artificial intelligence type of system. I think if you try and propose that you're going to replace the pathologist, you're fighting a losing battle, and I don't think you're going to, because ultimately you have to convince them. So I don't think that's a particularly useful strategy. But certainly, systems that prompt them to look at areas of interest that are really, and the point is not to really beat the pathologist. It's just to create better consensus across pathologists. And hope that the consensus is closer to the truth than any individual pathologist is on their own. Because ultimately, like in application, right? Like when someone gets, or when someone who has a heart transplant is getting this biopsy read, one pathologist reads it, right? They don't have a panel of expert pathologists reading the pathology. One person is doing it. And so the lack of agreement is concerning from that perspective. So AI systems to improve consistency or inter-rater agreement, absolutely. To sort of replace pathologists whole cloth, probably not. It is a bit of to replace whole cloth, it is a bit of a circular thing, right? Like, because you're going to rely on labels from them to train your model. So I think the best we can hope for is better consistency or better inter-rater agreement. Which would always be good. Yes.
So the next question is actually related to your last point, Casey. Is there any research to show the main reasons for the high inter-pathologist disagreement?
Oh, that's a good question. I don't know. Because they're doctors and are very proud and have their own, you know, a pathologist will have seen thousands of images, right? And I think that maybe this is the computer scientist in me, but the same way that a model is predicated on its training data, right? The data that it's seen, and can be influenced by the sort of vagaries of the training data that it's seen. A pathologist ends up having different focuses and different inclinations based on their own experiences and their own, and some of that is probably valuable and useful in the sense that they probably grow more sensitive to things that are more relevant to the populations that they see. And some of it is maybe just overfitting to the small set of slides that they ended up seeing in their career. Yeah. But I'm not aware of, I'm sure there are studies that have tried to answer that question. I just don't know.
Yeah, I think there was one very, very old paper, which obviously didn't cover all of this, but I think from the 1960s. But I think also some of the features like QWERTYs are very similar in terms of their pathology on the slide to rejection, which can cause, it's you see red and blue dots on a.
I don't,
not an easy
unless
thing.
you've seen a pathology slide and sat with a pathologist as they were reading, these things don't look like much of anything, at least to me. So, kudos to them doing as good as they do. We're just trying to develop tools to help them, really, right?
So we have to close, but I just wanted to have one last question. Somebody's wondering whether there is any racial data in terms of percentage breakdown of transplants globally, Canada or BC, and the rejection rate related to it.
So yeah, absolutely. There is ISHLT has reports that they publish periodically on those kinds of outcomes and demographics. I don't have the numbers on hand, but the ISHLT website is a good resource for that. I don't think they publish it every year, but every couple of years there's a report on outcomes that comes out that includes that information. Transplant BC would have that kind of data for BC specifically, if you were interested in that.
Yes, thank you so much, Tracy and Linda. I think the presentation that both of you provided today outlined quite a few things that I think data scientists need to look out for. Like phenotypes are very important. Asking the right questions are very important. Looking at the methodology behind, whatever you're applying, is that suitable for the data? And just being really thoughtful as to where the errors could come in and sort of looking out and then look at the results and figure out, does that make sense in the context? So thank you so much. There is obviously a lot more involved in both of your work than what you could present today, but I think it was a good overview.
Yeah, I tried to do justice in 45 minutes.
Yes, thank you so much. And thanks all of you for participating. Have a good rest of the day.